आई एम मुस्कान। आज हम फॉस्फोलिपिड पढ़ेंगे। बट हम फॉस्फोलिपिड पढ़ने से पहले लिपिड के बारे में जानेंगे। व्हाट इज लिपिड? Lipid term was first used by German biochemist Blohr in 1943. They are the ester of glycerol and fatty acid. And lipid are the heterogeneous group of compound related to fatty acid, include fat, oil, wax, and other related substance. Lipids are insoluble in polar solvent like water and ethanol, and soluble in polar solvent, fat solvent like benzene, ether, acetone, chloroform. Lipids are fuel reserve of the body. They regulate permeability of the membrane. They give shape and smooth look to the body, and they act insulating material and protect the internal organs. The classification of lipid. Blohr in 1943 has proposed the classification of lipid. The first class is simple lipid, the second class is compound lipid, and the third class is derived lipid. Simple lipid. They are the ester of glycerol and fatty acid. When one hydroxy group is esterified with one fatty acid and form monoacyl glycerol, and 2 hydroxy group is esterified with 2 fatty acid and form diacyl glycerol. When 3 hydroxy group is esterified with 3 fatty acid and form triglyceride या triacyl glycerol. This is a simple lipid. Fat and wax is a simple lipid. Compound lipid. They are the ester of glycerol, fatty acid, and other additional group like phosphate group and nitrogen base, etc. Derived lipid. They are derived from simple lipid and compound lipid by the hydrolysis. Second class is compound lipid. They are the ester of glycerol, fatty acid, and the additional group like a phosphate group and nitrogen base are is present. जो फॉस्फोलिपिड है वो कंपाउंड लिपिड के अंदर आएगा. कंपाउंड लिपिड भी दो टाइप्स के होंगे, फॉस्फोलिपिड एंड ग्लाइकोलिपिड. बट हमारा टॉपिक है फॉस्फोलिपिड, तो हम फॉस्फोलिपिड के बारे में पढ़ेंगे. Phospholipid is a compound lipid and they are ester with glycerol, fatty acid and phosphoric acid. Means phosphate group are present in the phospholipid. This is a phospholipid. Phospholipid are signal mediators, amphipathic molecules. They are involved in coagulation of blood cells. They are major constituent of cell membrane. They help in etc in mitochondria, and they play major role in transport and removal of cholesterol from the cell. They are involved in absorption of fat from the intestine. This is a function of phospholipid. Phospholipids are two types: glycerophospholipid and phosphosphingoside. Glycerophospholipids are four types: phosphatidylcholine means lecithin, phosphatidylethanolamine means ethylene, phosphatidyl inositol, and phosphatidylserine. Phosphatidylcholine means lecithin. They are widely distributed in nature and they are found in animal cells, and they are found in the nature. They are present in the soybean oil and yeast. Egg yolk is the rich source of the lecithin. The lecithin is play important role in fat metabolism in the liver. In the structure, they are phospholipid and they are made up of glycerol, fatty acid, phosphate group, and nitrogen base choline, and form phosphatidylcholine. But in the hydrolysis, they give nitrogen base choline, 2 fatty acid, glycerol, and the phosphate group. But in partial hydrolysis by the lecithinase enzyme, the lecithinase enzyme are found in snake venom. They cause removal only one fatty acid, and to yield substance is called lysolecithinase. Sorry, lysolecithins. This is a structure of lysolecithin. Phosphatidyl ethanolamine means cephalin. They are widely distributed in nature and animal cells. They are present in soybean oil, and the structure of cephalin is similar to lecithin. The cephalin is ester with glycerol, fatty acid, phosphate group, and nitrogen base ethanolamine. The serine is the biochemical precursor of ethanolamine. This is a structure of phosphatidyl ethanolamine. The cephalin is more acidic than lecithin. The cephalin is less soluble in alcohol than lecithin. Phosphatidyl inositol. This is a phospholipid and they are present in the brain tissue. This phospholipid is very important because they play important role in transportation process in the cell. They are ester of glycerol, fatty acid, phosphate group and inositol. Inositol is a hexahydroxy alcohol. They not contain nitrogen base. Inositol is present as the stereoisomer means myoinositol. They are present only the mitochondria. The mono, di and triphosphoinositol are also found, but in the hydrolysis of the phosphoinositol are they give glycerol, 2 fatty acid, 1 inositol and the phosphate group. The phosphatidylserine. It is a phospholipid and it is an acidic phospholipid. They are present in the animal tissue and serine is the biochemical precursor of ethanolamine. They are ester of glycerol, fatty acid, phosphate group and the serine and form phosphatidylserine. But in the hydrolysis of phosphatidylserine, they give glycerol to fatty acid, phosphate group and the serine. Phosphosphingoside means neutral phospholipid. They are different from other phospholipid because they not contain glycerol. They contain another nitrogen base sphingosine and the choline in the place of the glycerol. They are present in the nerves tissue and myelin sheath of the nerves. They lack in plants and microorganisms. In a syndrome called Niemann-Pick disease, sphingomyelin are stored in brain in large quantities. The sphingomyelin is electrically charged molecule because they contain phosphocholine as their polar head group. The sorry. The sphingosine carry phosphate group on its primary alcohol group and the fatty acid by the amide linkage on its primary amino group. This is a structure of sphingomyelin. In the hydrolysis of phosphosphingoside yield fatty acid, phosphate group, choline and sphingosine, but no glycerol. Thank you.